we start our presentation, I wish to acknowledge the people from the Kulin Nations on whose land Monash University is positioned. I pay my respects to their elders, past and present. Okay, just a housekeeping item. Please ask your questions for the speakers via the Q&A button at the bottom of your Zoom window. And these questions will be answered at the end of the speaker's presentation. So the chat section has been disabled, so questions must be sent through via the Q&A. Okay, so now it's my great pleasure to introduce our speakers today. We have two excellent speakers. Not only they are international leaders in the field, but also coming from home at Monash. So our first speaker is Professor Alex Fornito. Alex is with Turner Institute for Brain and Mental Health at Monash. So he's the head of brain mapping and modeling research program at the Turner Institute. So Alex is really a highly cited researcher in this field. He has won numerous awards. I won't list them here. So he's a world leader in developing new imaging methods to map human brain connectivity and applying these methods to investigate brain functions in both health and disease. So including characterizing brain connectivity in psychiatric disorders. Our second speaker is Dr. Stuart Oldham. Stuart is a research fellow at Turner Institute at Monash. He's also a research officer at the Murdoch Children's Research Institute. So his research interests including characterizing the organization of human brain networks with a particular focus on how this organization develops using advanced neuroimaging and the computational tools. So welcome both. I will now stop sharing my screen and then hand over to you, Alex, for your presentation first, for your introduction. At the end of your presentation, if you could pass to Stuart. So thank you both. I'm looking forward.
Thanks very much, Jolene, for that introduction and for the invitation to speak today. Is that working? Can you all see my screen? Great.
Excellent.
And thanks everyone for tuning in. So today we are going to talk about some work that Stuart did during his PhD in our lab, focused on trying to develop generative models of brain network wiring. And I don't want to take up any longer than I have to. I want Stuart to really go into all the wonderful work he's done, and he's got much better presentation style than I do. So I will begin with just a brief background to set up the general problem and area, and then I'll hand over to Stuart, who will go into detail about what exactly he's done in his work and how he's gone about tackling some of the problems that I'll explain. So really the work today is motivated by a long-standing interest we have in our lab in trying to understand the underlying principles of brain network wiring. And an important concept in this space is that of a connectome. So a connectome the term was first defined by Alex Fornito and colleagues in 2005 as a comprehensive structural description of the network of elements and connections forming the human brain. So it's essentially a wiring diagram of the brain, and we can construct connectomes at lots of different scales, from the level of individual neurons and synapses through to axonal fibers linking populations of neurons, and then the macro scales we typically deal with human imaging. It's really looking at how large brain regions are connected by bundles of axonal pathways. And this is the scale that I'm going to focus on today, this macro scale. So we're really talking about models of human macro scale brain connectivity. Now, there's pretty well established pipelines for using different types of MRI for mapping different types of connectomes. The basic idea is that we divide the brain into different areas, and these act as our network nodes, shown here. And then for every pair of regions, we map some aspect of structural or functional connectivity. I'm going to focus on structural connectivity today, so we're really talking about the anatomical connections between regions. So we do that for every pair of regions, and then we can represent that connectivity in the form of a matrix, where each row and column represents a different brain region, and each element encodes the type of the type and strength of connectivity between regions. And once we have this matrix representation, we can generate a graph model of the network. And once we have this, we can then do two broad classes of analysis. So one focuses on connectivity, so looking at variations in the type and strength of connectivity between regions. And the other focuses on topology. And topology is really concerned with how connections are organized with respect to each other in the network. And this is where we really use the mathematics of graph theory to derive a number of different quantities that try to explain or describe how these connections are organized. Now, there's been a lot of work that's gone into trying to understand the key topological properties of brain networks. And you know, the basic idea is that the connections in brains are nearly arranged completely at random, nor are they completely regular. So they kind of have features of both, which gives rise to what we call a complex topology. So there's a lot of interesting structure in there. And so the question is, well, where did this structure come from? How did it arise? What are the underlying constraints on brain network wiring that could lead to a network that looks like this? And some of the most general and long-standing principles were first proposed by Santiago Ramon y Cajal over 100 years ago, when after spending countless hours peering through the microscope and studying the structures of neurons, he concluded that all of the various conformations of the neuron and its various components are simply morphological adaptations governed by laws of conservation of time, space, and material. So in terms of conservation of space and material, he's really talking about a pressure to minimize metabolic expenditure wherever possible. So larger brains consume more energy, brains with more cells or other types of physiological material consume more energy, and generally physiological systems try to minimize metabolic expenditure wherever possible. In terms of conservation of time, he's really speaking about the need for a nervous system to communicate rapidly and effectively. And that has obvious implications for the survival of an organism. We need to be able to respond to what's happening in the environment quickly and accordingly regulate our own internal states. Now, a lot of work has gone into understanding the roles of these principles, particularly the conservation of space and material. And a number of studies looking at different properties of brain organization, whether it's the spatial arrangement of individual neurons or large-scale brain areas, or the branching patterns of dendrites, or things like the fraction of gray and white matter within the cortex. All of these properties have been explained in terms of wiring cost optimization. So you can think of wiring cost as referring to the overall volume of wiring required to connect a neural system. And this has really been used quite often to operationalize this conservation of space and material idea. So the idea is that networks with lower wiring volume require less space and less material and consume fewer metabolic resources. So there's been a lot of interesting work suggesting that a lot of aspects of neuronal organization are consistent with an optimally wide or minimally wide system. But a few years ago, in this really nice study by Marcus Kaiser and Klaus Hilgertag, they showed or presented evidence to suggest that nervous systems are probably not optimally or minimally wide in an absolute sense. So they took a mesoscale connectome from the macaque based on track tracing data and a neuron and synapse connectome of C. elegans, and they maintained the connectivity pattern between each node, but rearrange their spatial positions and ask, well, is it possible to find a different spatial layout that is associated with lower wiring cost? And they found that it is indeed possible to find these configurations. So in blue, we have the wiring cost of the original network, and in red, we see the cost of the minimally rewired network. So you can see there's quite dramatic reductions in wiring costs that are possible, suggesting that brain networks are not absolutely or minimally wide. But they found that these networks had a longer average path length. So the average path length is the average number of nodes it takes to get from one point in the network to any other. So you can think of this as being related to how quickly different parts of the network can communicate to each other. If I can go from one area of the network to another in just a couple of hops, then my communication is quite rapid. And really, average path length you can think of as providing an upper bound on how quickly the network can communicate. You can see in these cases, the empirical data has a lower average path length than the rewired networks. So this points to a bit of a trade-off. On the one hand, you can minimize wiring costs, but it comes at the expense perhaps of conservation of time. And this is actually, if you think about it, implicit in Cajal's conservation laws. So if you ask, well, how could I configure a network that would optimally conserve space and material? And the network would look something like this. Each node would connect to its nearest spatial neighbors. So the overall volume of wiring would be low. But if I want to send a message from the front of the brain to the back, I need to go through lots of intermediary connections to do so. So it's quite a slow, inefficient way of communicating. If you then ask, well, what does a network look like if I want to optimally conserve time? It can be shown that a random network does a pretty good job of doing that. And so you can see you get these long-range connections that cut across the network and dramatically reduce the average path length, but they do increase the total wiring volume. So on the one hand, we can conserve time, but not material. On the other hand, we can conserve material at the expense of time. Now, brain networks are somewhere in between these extremes. They have lots of short-range connections, so that conserves material, but they do have a fraction of strategically placed long-range projections that act as shortcuts in the network and help to conserve time and also provide a lot of other functional advantages. So we really appear to have this trade-off here. And so there's been, over the last 10 plus years, there's been a lot of work that's been trying to model the role that these trade-offs might have in shaping brain network wiring and explaining some of these complex organizational properties of connectomes. One of the simplest models has this form here. So we have the probability of connecting any two nodes, U and V, is proportional to some exponential function of the distance between them. So this is really capturing wiring cost. So the idea is we're not trying to minimize wiring in an absolute sense, but we are saying that there is a cost associated with each connection. So the longer a connection is, the less likely we're going to form it, but everything else is at random. So in this model, we grow networks computationally, connecting nodes at random, subject to the constraint that long-range connections are less likely to form. And this free parameter here, eta, determines the extent of the distance penalty. So here you can see this is a roughly exponential decay in the probability of connection as a function of the distance. And we can play around and look at different values of eta, and they can result in either a more stringent distance penalty or a more relaxed distance penalty. And so eta is a parameter that we fit to the data. So we essentially try lots of different values of eta and work out which one best fits our empirical properties. Now, this very simple model actually goes quite a long way in explaining diverse features of brain organization. So it's been used to show that just using this very simple wiring rule, you can generate networks that have similar degree distributions and motif spectra to track tracing connectomes in macaque. Motif spectra, they're just different ways of connecting subsets of three nodes. It's also been shown in human data that this simple model can reproduce distributions of topological properties such as average path length, degree, clustering coefficient, betweenness reasonably well. And other models that sort of simulate the outgrowth of axons according to this kind of rule also do quite well in reproducing diverse features of the macaque connectome. And so the success of this model has really led some people to propose that this sort of exponential distance rule for neuronal connectivity might be a really important and fundamental property of brain organization. And this aligns with a lot of empirical data showing that generally the probability of two regions being connected and the strength of their connectivity and the degree to which their patterns of gene expression are coupled, all of these decline roughly exponentially as a function of distance between regions. Recent work, however, has compared these models to a set of models that more explicitly try to capture the trade-offs implicit in Cajal's laws. So the basic form of these models is something like this, where again, we've got the same, this part here, everything to the left is the same as what we've just seen. So probability of connection is proportional to an exponential function of the distance. But we also have this additional term here, t, which is a topological term. And the idea behind this term is it's designed to favor connections that are topologically favorable. So connections that are thought to provide some kind of functional advantage. So under this model, we're less likely to form long-range connections, which is given by the wiring cost. But if a connection is viewed as providing some kind of functional advantage, then we have a bias towards forming it. So it's trying to capture this idea between a trade-off of space and material and time or adaptive function. I'll point out that we're focusing on an exponential term on the distance here. Other people have tried power laws. The details are not so important, but we're focusing on an exponential distance penalty. So the question is, well, okay, well, what is functional benefit and what do we use for this T term? And so lots of different topological properties have been tried. The details are not so important, but in a series of studies, it's been shown that this specific property here called the matching index seems to win. So here this is work by Rick Betzel, where he's looked at model performance in reproducing topological properties of human diffusion MRI connectomes. This is an index of model fit, lower values indicate better fit. And so this matching index model is doing the best. And the matching index is essentially the proportion of nodes, neighbors that two nodes share in common. So the idea is if two nodes connect to other similar nodes, then they're more likely to form a connection to each other. And so this bias towards making those connections is coupled with this distance penalty. You'll also note that this purple box here corresponds to a purely spatial model. So that's just the exponential distance penalty without topology. So this result is suggesting that this purely an exponential distance rule alone actually performs the poorest when compared to all these different trade-off models. So suggesting that while you can get quite far with a simple exponential distance rule, actually, when you incorporate trade-offs into the model, you do a lot better. So we might say, okay, well, job done, that's fantastic. We've just used a whole bunch of fancy math to confirm what Cahal said over 100 years ago. And in some senses, some people might say that's trivial, but personally, I find that quite remarkable and satisfying that you can have just a very simple two-parameter model, and that appears to explain lots of different complex properties of brain organization. But there are still some caveats to the work that's been done so far that prevent us from really drawing a definitive conclusion on some of these questions, such as whether a trade-off model is absolutely required and whether it does in fact perform better than a purely spatial or distance-based model. And so with that, I'm going to hand over to Stuart now, who will go into those problems in a little bit more detail and then talk about how we've sort of tackled it in his work and some of the results that we've found. So without further ado, I'll hand over to Stuart.
Thanks, Alex. So in resolution to the shortest cliffhanger ever, I'm going to be expanding on what Alex was mentioning. So as he was saying, there are a number of sort of issues looming over these results with generative models. And my work has been to explore these issues and try and provide different solutions to them. So there are four main issues which I was trying to address in my own work. And the first concerns this idea of the wiring rule formulation. So as Alex was showing before, that we define the wiring rule as some probability dependent on, is defined by some exponential function, the distance between two nodes, multiplied by their topological value. Now what I'm showing in this plot is what you get for different values of topology. So we've got all these different values of distances, and I'm showing what the probability is if you've got a topology of 1, what the probability is for a topology of 1, if you've got a topology of 1.5, and if you've got a topology of 3. Now what you can see is that these effects are non-linearly distributed. In fact, they're mostly concentrated on short-range connections. So what this means is that as we're increasing the value of topology in these models, these effects are only being concentrated for short-range connections and aren't affecting long-range connections. Now, our understanding of a trade-off model should be that an edge which offers is sufficiently functionally valuable should be able to overcome its distance penalty. But under this multiplicative form, it's very difficult to do so. You either need to have an outrageously large value of topology or use some huge weighting. So to address this, we came up with an additive formulation. So whereas before we were raising topology to some power, now we're adding in these two terms, the distance and topology term, with an alpha parameter controlling the weighting and contribution of topology to the overall probabilities. And this model, we see a different result. So now when we're increasing the value of topology, we are able to assign more weight to these long-range connections. So you can see that as the value of topology increases, that now a long-range connection can be more likely. And so we wanted to investigate to see whether this alternative formulation could offer an improvement over the commonly used multiplicative case. Another issue concerns biological plausibility. So as Alex was showing, that a lot of these generative models use topological measures. And these topological measures are somewhat abstract and they're hard to link to underlying physiological mechanisms. So what we aimed to do was create a generative model which can incorporate more realistic physiology and developmental events. And one of the key developmental events we thought we should include was the changes in cortical size and shape. So when the brain is developing, it is obviously much, much smaller. And it is at these same time points that connections are being formed. In particular, it seems as though most axonal formation is occurring in the second half of gestation, particularly between 20 weeks and birth. And so if we could capture how the brain is physically changing in size and shape, then we can incorporate that into our model. And so why might that be important? Well, we were just talking before about how there is a distance penalty. And so it might be because the brain is physically smaller that this penalty is relaxed, and so it may be more likely for long-range connections to form or other connections which may not form in adult data. So to do this, we were lucky enough to obtain fetal MRI scans, which are taken of babies while still in the womb from 21 weeks gestational age on a week-by-week basis to 38 weeks gestational age. And we wanted to incorporate this data into the model. And so the basic idea is because we want to measure changes in distance, we need to know where nodes are across each of these time points. And we have our parcellation data in the adult brain, which gives us this information. So how do we transfer it to each of the fetal brains? Well, we use a technique known as multimodal surface matching. So with multimodal surface matching, you can project features onto the surfaces of two brains. In this case, I'm showing sulcal depth. And then you inflate the brains to a sphere. Now, these are surface meshes, and they're a triangular mesh. And so what they are is they're made up of vertices. So if you zoom all the way in, and each of these vertices contains information about the sulcal depth at that point. And the basic objective of multimodal surface matching is to move around these vertices along the surface of the sphere to align the data on one surface to that of the other. So you can see here that data on one surface is being sorted to roughly match that in the other. And the cool thing about this is that the end result is you get vertex-to-vertex correspondence. So you know where one point in one brain would be on another brain. And so we can take out the adult data and project it into, for instance, the 30-week brain. And as I mentioned before, this is useful because we have our parcellation data projected onto the adult. And so what we can then do is we can now project this back through time across each of these individual time points and then map the distances between nodes throughout. And the basic way we incorporated this into our model is we start with the distances at the earliest time point, form some connections, move to the next time point, use those distances, form some more connections, move to the next time point, so on and so forth. And we call this a growth model. We also have a static model, which is what people have previously used, where we just take the adult distances and perform the calculations on those. Another aspect of physiology that we're interested in is that over the last 10 years, there's been increasing evidence suggesting that regions with similar cytoarchitecture are more likely to be connected with each other. And this has led to the proposal of the architectonic type principle or structural model. It's also been shown that regions with similar gene expression are more likely to be connected. But largely, these processes haven't been incorporated into existing models, so we sought to do that in our work. So first up, we're looking at correlated gene expression. So this is taken from the Allen Human Brain Atlas. And basically, this was 6 samples, and they measured gene expression all across the cortex. And it's extensive pipeline at the end of this is we're able to measure the gene expression for a given region. And what this gene expression shows the expression of genes in the brain which have shown elevated expression relative to the rest of the body. And so you get this for every single possible combination of regions. And then the basic idea is we want to compute how similar any two regions are in their gene expression, so we basically just correlate them. And then we can repeat this correlation procedure for every single possible set of regions, and we get this correlation matrix. And an issue with this correlated gene expression is it shows a distance dependency. And so what I mean by that is that we see that regions with high correlated gene expression, so regions that are more similar in their genetic expression, tend to be closer together than those regions that are further away. And typically what is done is we want to see an effect beyond just a simple lower order spatial order correlation. So we want to correct for this. But there is some evidence suggesting that even in this uncorrected form, there may be some meaningful physiology behind this spatial order correlation. So we want to include this as well. But the way we correct for it is we fit a distance trend and then we remove that from the data. And this results in us getting corrected correlated gene expression, or CCGE. Another measure of physiology relates to the cytoarchitecture, as I was mentioning before. And so the way we extract this is with what's known as microstructural covariance. And the way this works is we define the outer gray matter surface and the white matter surface. And then we construct a series of equivolumetric points between each of these. Equivolumetric surfaces between each of these. And the basic idea is we want to sample along each of these surfaces the underlying intensities of the MRI image or whatever other image that they cross. And so for two given regions or any given region, we basically sample each of the points along each of these surfaces and average them. And this results in us obtaining a vector of the average value along each of these surfaces. And then similar to before, we can just compute the similarity between these two values for every single pair of region and construct a matrix of this. So the first way we did this was using histology data from the Big Brain project. And so this is roughly an index of cytoarchitecture. And we also used the T1, T2 ratio as defined as extracted from the human connectome project. And the T1, T2 ratio is thought to be an index of myelination. So histology and T1, T2 are both considered measures of underlying psychoarchitecture. And so at the end of this is we have 4 different physiological constraints. So we have uncorrected correlated gene expression, corrected correlated gene expression, multivariate profile covariance for histology, and the same again for the T1, T2 ratio. And the way we incorporate this into a model is pretty simple. So we can either assign the probability as being equal to some physiological constraint raised to some power, and we refer to this by the name of the particular physiological constraint. What we also wanted to see is how this may interact with distance, because we've seen wiring cost is quite fundamental. So we just include this by simply just replacing topology. The next issue that we wanted to look at concerns spatial properties. So Alex mentioned a little bit before that how we evaluate the fit of these models, and I'm just going to expand on that a bit. So the way we define fits in these generative models is to compare statistical distributions of topological properties. So for instance, we have our data and we have our model, and we might compute the degree distribution and the edge length distribution in each. And then for each of these, we can compare these distributions directly by converting them to a cumulative distribution function. And so it's not just degree and edge length that we use, we also use betweenness and clustering. These are all somewhat fundamental properties of brain networks. And basically the way we measure our fit is we look across all of these and we look for the greatest discrepancy between these two curves. And that becomes our index of model fitness. However, a brain is more than just statistical distributions. The spatial embedding of topology is important and a key feature. So for instance, if we consider high degree nodes or hubs, we know their location is very important and is consistent, but we don't really know how well generative models do in actually replicating this kind of spatial embedding. As you can see here, even though these two networks are quite similar in their distributions, they have a very different patterning of where their hubs are. So potentially these models may be doing very well at replicating the statistical distributions, but maybe perhaps not the spatial embedding. The final issue that we sought to address was this idea of cross-validation. So these are models and they involve fitting of parameters, and there's the potential risk that there may have been some overfitting. So to address this, we developed a cross-validation procedure. So the way this works is you do some optimization, you sample all through this parameter space, and eventually you find the set of parameters that gives you the best result. And for our cross-validation procedure is we find these optimal parameters for every single subject, and then we apply them to a given subject's network. So for instance, here we're taking subject 1's network and we're applying the optimal parameters for subject 2, the optimal parameters for subject 3, 4, etc., etc., etc. And we repeat this for each set of parameters 10 times, so you get a distribution, and then within each of these sets, we find the average fit you would get, and then we average across these again. And this gives us our cross-validated fit for each individual participant's network. So they're what we were addressing. And so to test this, we performed generative modeling in 100 structural connectivity networks constructed from the human connectome project. So the first thing we did was the additive and multiplicative models. What results did we find? So this is what we found. Now to interpret this plot, on the y-axis is the cross-validated fit. So this is a lower value indicates that the model more closely replicated the brain networks, and each bin on the x-axis contains a different topological model. And the additive form is shown in the dark outline, and the multiplicative form is shown in the dotted outline. Now, what we can see that in many cases that the multiplicative is performing better than the additive. However, if we look at the best possible case, which is the matching, we see that the additive model is offering a sizable improvement over the multiplicative case. And so this was good. It suggested that our additive formulation is working. But one thing we were wondering is, with our additive formulation, it's expressed as just all that's happening is topology. It's just having some global scalar applied to it. But potentially we're thinking that maybe it might require some nonlinear weighting in order to properly work, particularly for these other topological values where we won't see any improvement over the multiplicative case. So we altered this into what we called an additive nonlinear formulation, where now topology we're raising to a power gamma in addition to having this alpha parameter. And when we compare this model to just the purely additive form, what we see is that, again, we see most cases that it shows some improvement. And particularly if we look at the matching case, again, we see that our nonlinear form offers a small improvement over the simple additive form. And so with this information in mind, all results presented here on out will just use this particular form shown below. So the next result concerns our static and our growth models. So again, these are similar to what the plots you were seeing before, but now the dark line is showing growth and the dotted line is showing the static case. And so what you can see is that in many cases, the growth offers a small advantage. Now, one thing we're thinking of, and which Alex was alluding to at the start, was whether a simple spatial or geometric model, which is only using the distance term, if we could incorporate growth, potentially could that offer a substantial improvement? Could we be able to capture long-range, valuable connections that may not be able to capture in a simple static case? And our results say we see a small slight improvement. However, we see a much larger improvement when we look at the matching topological model. So this model is really showing an extra benefit to including growth. And so one thing we're wondering is, well, why is the difference for most other models only quite small? And the reason is if we actually look at the actual parameters themselves. So here I'm showing the relationship between probability and distance, similar to what Alex was showing before. And so I'm showing this for what we saw for static models and what we generally saw for growth models. So you can see for static models that the decay isn't as strong as it is for the growth model. So this says that in the growth model, there is a stronger distance penalty. And this sort of makes sense because because the growth model, the distances are going to be smaller than the static case, which again, to remind you, is based purely on the adult. And so the model is trying to compensate for these overall smaller distances by simply imposing a stronger penalty. And this, to a certain extent, largely negates the advantage of having a growth form. Next was the results of the physiologically constrained models. So here I'm showing the results. So again, we had the 4 types. So we had corrected correlated gene expression, uncorrected correlated gene expression, microstructural profile covariance for histology, and the same again for the T1, T2 ratio. And we also took the matching model because, as we had seen before, it was the best performing model, and we compared their performance. Now, what struck out to us was the first thing is that we saw when we used correlated gene expression in its corrected form just by itself, it offered the worst fit. It wasn't replicating the data well at all. However, when we combined it with spatial information, we saw a dramatic improvement. And further still, we saw that when we used the uncorrected correlated gene expression, and just to remind you, the uncorrected correlated gene expression was not corrected for the distance trend, that these results show the very best result. And the most interesting thing I find is this uncorrected correlated gene expression model. So this model isn't including a separate spatial term where we're combining it explicitly with distance. So this is just purely done on the correlated gene expression data, and it's providing almost the very best fits. And so this very simple one parameter model can outperform almost everything else, including the abstract topological measures we'd seen before. We can also see some of our physiological constrained models also do quite well. So the microstructural profile covariance for T1, T2, that also is doing on par or even better than the matching case. But this is then followed by the histology data. And finally, we want to consider the results with the spatial embedding. So we've seen that these models can generally do okay at replicating the statistical distributions of topological properties, but how do they go with the spatial embedding? So first off, I'm starting with the topological model. So this is with the matching model. And as you can see, this is the cumulative distribution function. So this is what we're using to define the model fits, remember. And you can see that the empirical data is shown in black and the model data is shown in blue. And you can see, and you probably agree, that these are quite close fits. However, if we look at the actual spatial embedding, we see that it's not quite so rosy. So for this, we're looking at the actual degree of each region in the empirical data and the actual degree of that same corresponding region in the simulated model data. And you can see that we actually get a weak negative correlation or no correlation at all. And you can see if you actually map where the degree maps, you can see that they're very different. So in the empirical data, we sort of get distributed hubs based in insular and prefrontal and temporal areas, whereas with the model data, that degree tends to sort of concentrate in the parietal lobes. So this isn't very good. And if we then look at clustering, which remember is another thing that makes up our model fit formulation, we also see it's not doing very good. And you see the same if you look at betweenness, or if you look at the mean distance of every given region. So this is all telling us that existing models haven't done a particularly good job of replicating the spatial embedding of topology, which again is a key feature of brain networks. So what about our physiological models? We saw they could improve the fits. Can they do better with the spatial embedding? And the short answer is, yeah, kinda. So again, we can see here that now we're getting a correlation between the degree of the empirical networks and of the model networks of about 0.2. And so this represents an improvement and definitely steps in the right direction. For the other topological properties we're interested in, the results are a bit more mixed. For some of them, like clustering between us, this model still fails to replicate. But for things like the distance distribution, it does begin to replicate the spatial topology of that as well, quite much better than we've seen previously. So on to our conclusions. So generative network models, including growth, offer a small but still significant improvement over non-growth models. And so likely we think the reason that for this is that when the brain is smaller and combined with topology, is that this allows for longer range connections which may not otherwise be able to form as a result of the severe distance penalties that they're exposed to in the adult brain to then be able to actually be included into the model. We also see that our physiologically constrained generative models show more promise in offering a more biasly grounded model. So we saw that they improved the fits of our generative models. So why might this be? What's the potential mechanism explaining why these physiological models offer improvement? Well, gene expression, histology, and myelination all roughly relate to this idea of laminar structure or cortical layers. So, most people are familiar that we've got the 6 cortical layers, and the structure of these varies throughout the cortex. And an idea proposed under the structural model is that the similarity in the cytoarchitecture or laminar structure reflects differences in developmental time courses. So regions that develop at have a similar developmental period will tend to form connections with each other. So potentially these are what our physiological measures are tapping into, this idea that regions with similar developmental time course are more likely to become connected. And the final main takeaway from this was that generative models fail to reproduce the spatial positioning of hubs and other topological features. So we're still not 100% sure why this might be the case. One potential thing is that because a lot of these models are relying on distance, that this distance provides is just a huge constraint and tends to result in things like the hubs being located in regions which tend to be the most spatially close to everything else. And so what we need to do is find some generative model which can really fully overcome this kind of distance penalty to properly form the spatial embedding of brain networks. So that's where I'm going to leave it. I'd like to say thanks to Alex, as well as Ben Fulcher and Kevin Aquino for really helping to shape and conceptualize the ideas behind this project, and also to Irina Arnikevich-Ute, Rosa Shishigira, and Casey Picola for all their help and assistance in getting the data ready for this project. So thank you for listening.
Excellent. Let's applause to our speakers. Thank you very much. Very insightful presentations. Now we have a few minutes for questions. I think we have time for 2 to 3 questions. So there's already a couple in the Q&A. So there's a question referring to, I guess that's the gene and the connectivity. It could be a case of chicken. So the question, this first comment, it could be just a case of the chicken and egg issue. So do you reckon it's just the genes that's influencing connectivity, or the connectivity that dictates the similarity in gene expression? So I think the questions are raised from during your presentation, Stuart. Do you want to take a shot?
It's a good question. I guess my overriding assumption would be that it's genes which are shaping connectivity. I mean, I guess one issue is we're sort of looking at a more macro scale sort of level of connectivity where it can be, I guess, tricky to sort of conceptualize how these sort of gross axonal fibers may be shaping individual expressions of genes within each region. It's a good question, one which I haven't really thought about, but I guess our overriding assumption that it's primarily one way. And Alex, do you have anything to add?
Well, yeah, I mean, I think it's a good question. So, I mean, the gene expression measures are taken from the adult brain, which is sometime after when connections are actually formed, and it is possible that transcriptional patterns are different at those stages. Some genes show persistent patterns of expression through development, others don't. So obviously, in an ideal world, we would have gene expression at each stage of development. But at the moment, there are some atlases that try to do that, but they're not as anatomically comprehensive, which makes it hard to do the whole brain modeling. It is possible that the gene expression similarity is driven by a similarity in cytoarchitecture. Because the gene expression measures are taken from microarray, they're going to be sensitive to regional differences in cell types and things like that. And so maybe the gene expression is just a different measure of cytoarchitectonic similarity. So it's in a sense complementary to the T1, T2 and the big brain data. But also contain, regions could have similar gene expression for other reasons. And so, yeah, we can't be 100% sure about what's exactly driving it.
Excellent, thank you for the answer. So next question is comment, is great talk, and thank you. Could we improve the matching of the spatial distribution of the hub regions by using a correlation-based fit measure instead of the KS stats? So the KS statistics doesn't care about relative ranking between the regions. So is that correct? This question from the audience.
Yeah, so I can answer that. So yes, it's true. The KS doesn't care about the relative rankings between regions. It's a normalized measure. So what Kaya was saying is, like, could we change our model fit to instead look at the actual correlation between the values itself? And yes, in fact, we have previously done that. And we find that it really didn't improve these correlations much at all. And furthermore, that for these, when we did this optimization to produce the best correlation, the actual fits, as we were previously doing, of those model networks were complete mismatch to the actual underlying data. And I've also investigated very comprehensively sampling the entire parameter space that we're searching through and seeing what kind of correlations we get. And at least with our data, we've never been able to see any correlation consistently above 0.3. So even if we change our optimization, we don't seem like it's going to improve the fits.
Okay, great, thank you. There is a comment / question from Murat. Awesome work. I was curious in your growth model whether or not you took into account the influence of the intracranial volume. Reason being that intracranial volume is a clear physical barrier constraint for the brain. So cycle and gyral folding, which in turn affects connection length and topology. Question from
Well, the short answer is no, we didn't directly include the influence of intracranial volume. I mean, the other thing to note is that for our growth model, this was sort of a group template. So it was only based off some group average MRI scans that were made available. So we weren't able to assess anything like the variability in intracranial volume across different subjects. So no, we weren't able to assess that.
Okay, great. Well, thank you very much. I think that's conclude our session today, and we have excellent, very insightful presentations. Thank you, Stuart, and thank you, Alex. So let's conclude our session. Before you go, we have our magnetic particle imaging virtual lounge and webinar on Tuesday, 15th of June from 3 to 5 p.m. So registration details are on the MBI webinar page and also our social media. So thank you, and thanks everyone for attending this seminar. Looking forward to see you again next time. Thank you. Bye.